Good afternoon, everybody. It's with extreme pleasure that I have the privilege to introduce my friend Spencer Jones. Spencer, born and raised in the Amador foothills around Placerville. He then went on to receive his bachelor's in geology from the University of Oregon. But he really found his passion for wine when his family started Iron Hub Winery back in 2014. And after working a few harvests with his dad, who also attended and graduated from the Masters of Science program in Davis, he decided to forego pursuing a master's in geology, which he was originally planning on going into the petroleum industry. But thankfully, he decided to join us here at Davis. Last year, he interned at Bennett Lane Winery as part of his program here. And after graduation, he plans on returning to Iron Hub, where he has been managing the vineyard for the past 4 years. And he's going to continue doing a fantastic job. Without risking being a little bit editorial here, the wines at Iron Hub are absolutely fantastic. And if you haven't had a chance to go up there, I highly recommend it. And it's been a real great pleasure to know Spencer. So without further ado, take it away.
Right on. Thank you, Marco. So the title of my literature review is a comparison of rootstocks in potassium accumulation and evaluation of petiole potassium content as an indicator for berry potassium content. So the outline for my presentation: first, why is potassium accumulation by rootstocks important? How is potassium currently measured for grapevines? What are the seasonal trends of potassium accumulation in the grapevine and berry? What is the importance of root hairs for potassium accumulation? What are the known mechanisms of potassium uptake by the root system? Is there a consistent effect of rootstocks on potassium accumulation in the petiole and juice? And lastly, is petiole potassium content an accurate indicator for berry potassium content? So potassium has a strong influence on the wine quality. There's a strong correlation to the potassium concentration and the pH of a solution. So here below, you can see, can you guys see my mouse? Very cool. So you can see Chardonnay and Syrah, and this is potassium concentration in millimoles per liter. And you can see as the concentration of potassium increases, so does the pH of the juice. So this correlation has a strong effect on the microbial stability of the wine, the color quality, and then lastly, potassium bitartrate precipitation, which has a huge effect on the perceived consumer quality. So overall, the goal is to reduce potassium content in the berries for vinification, and we're going to see if that can be done by rootstocks. So potassium, the measurement method for evaluating this began back in the 1920s. They needed a method to assess the soil nutrient conditions in a vineyard. So then they began to measure the vines to assess the soil nutrient conditions. And in 1956, Cook reported measuring the petiole crossum inflorescence during anthesis as the most accurate method for measuring the soil conditions and the vine's needs of nutrients. And then in the mid 80s to early 90s, they began to notice that the pH in Australia of the wine was increasing after the use of rootstocks. So Rule, through 1989 through 1993, did a lot of work with rootstocks and potassium uptake, and he reported a strong correlation of the petiole potassium concentration at anthesis to the pH at harvest of the juice. So this began, in science for several next decades as the primary method to measuring potassium accumulation in the vineyard. So now I'd like to discuss, the potassium accumulations throughout the growing season. So during the winter, the vine will store between 30 to 65 grams, 65 grams of potassium in its perennial storage. And then this is, however, minor. Over 90% of potassium needed for the vine is actually sourced from the soil. Potassium is transported from the roots through the xylem and in the phloem up into the berry and the canopy. Near veraison, that's when the phloem becomes the only primary transportation for potassium. So this represents the switch from symplastic unloading to apoplastic unloading, allowing for high concentrations of potassium to build up in the berry. So the berry is the primary sink for potassium. An average about 29.8 pounds of potassium per acre are absorbed from the vineyard. And so here in this diagram, this is the potassium content in grams per vine, and then the days following bud break as the x-axis. And so you can see following bud break, how the concentration in the trunk and in the roots, how these both lines are declining. So those are providing the initial potassium needed for bud break. And then the remaining potassium begins to absorb from the soil. And then at flowering, you can see how potassium concentration really begins to increase, especially in the bunches once they're established. And then so you can see at veraison, one thing that's really interesting is, where's my mouse? I'm losing it. There it is. Okay, so you can see how the leaves after veraison actually begin to decrease in concentration of potassium. And so this is potassium being remobilized from the canopy going into the berries. And then at harvest, obviously the grapes are taken away, but you can see potassium actually begins to still increase. And this is replenishing the roots and the trunks. You can see down here below how the concentration begins to increase, and that's in preparation for the following growing year. So potassium is considered the major cation in a grape, and it's also considered the most depleted macronutrient at the end of a growing season. So potassium is a significant component of biochemical pathways in vegetative tissue. It contributes to osmoregulation, turgor maintenance, photosynthesis, cation balance, and plant signaling. And so here's an example of guard cells of the stoma and on a leaf. And you can see how potassium is pumped in and out of these guard cells, which results in the inflow of water and outflow of water, which opens and closes the guard cells. So this is just one example of many of what potassium contributes to the vine. So now I'd like to discuss the root hairs for potassium uptake. So the root hairs can absorb potassium ions in the soil either through root growth interception, mass flow of potassium ions driven via transpiration, or diffusion of potassium ions from high concentrations to low concentrations. The radius of these fine root hairs are 5 micrometers or less, so they're very small, able to grow into the small pore spaces of the soil to retain the ions and other nutrients. And the actual zone of the fine root hairs right behind the tip is termed the maturation zone. That's where the ions are actually absorbed. And so the rootstock morphology of root hair branching and bulk density varies significantly based on the rootstock. And yes, there are general trends for certain rootstocks being more vigorous than others. However, if you actually look across literature, there are very few conclusive rooting patterns of rootstocks across varying site conditions. And so here in this photo, you can see how it looks about a foot down in the soil. There appears to be a hard pan, and a lot of these roots are starting to grow horizontally just because they're no longer able to penetrate deeper into the soil. So the soil has a huge effect on the root pattern and root morphology. So continuing on with these, the root hairs for accumulation. So one study correlated the potassium accumulation to higher total root length and higher root surface area. 10114 had the highest potassium concentration in the petiole and also had the highest % of fine roots. So here, this is just, these were grown in a greenhouse in pots, and you can see how 10114 has a high density of fine root hairs compared to like Ramsey. So, but like I said, this is just, obviously this is in a greenhouse setting, not actually in a vineyard. So now I'd like to talk about the mechanisms for potassium uptake. So potassium ion channels and transporter proteins is what allow the roots to absorb the potassium ions and transport through the plasma membrane. So potassium ion channels are considered low affinity, so they passively diffuse potassium ions, and these are active when external conditions of potassium are high in the soil. So they contribute to potassium uptake and also potassium transportation. Right now in grapevines, the shaker channel circ has been identified in the berry pre-veraison. And then also another shaker channel, VVK1.1, has been active in the phloem and in the pulp throughout the growing season. And then for transporter proteins, these are considered high affinity. They require energy. And so these would be activated when external conditions of potassium are low in the soil. So VVKUP1 is one transporter that's been cloned in lab, and its active pre-veraison in the vine. So one thing that's really interesting, our understanding of these mechanisms actually does not come from studying rootstocks. It's actually coming from Arabidopsis, which is for plant biology the most understood, well-known plant. So by understanding that plant, we've been able to identify genes and correlate them to the ones in vines. So we're still in our infancy for understanding these mechanisms and actually what might be triggering signals which are resulting in potassium uptake and then transportation within the vine. So now I'd like to discuss, when I first began this literature review, I really wanted to focus on rootstocks that always had higher potassium accumulations or lower potassium accumulations. And so for instance, Vitis champagnii, that appears to increase potassium uptake. That's like the rootstock Dogridge or Ramsey. Several studies have noted this rootstock to increase potassium uptake, whereas 140RU has appeared to restrict potassium transportation in the roots. So it accumulates the potassium in the roots. However, something is restricting the potassium actually being transported up into the canopy. However, overall, there's significant variation of the rootstock's potassium uptake in literature. For instance, Coder in 2010 reported 1103P and 10114 accumulating the highest potassium concentration in the petiole, whereas Gautier in 2020 reported 1103P and 10114 accumulating the lowest potassium concentration. So this huge variation in these across literature is due to greenhouses settings versus actual vineyards, whether the rootstocks are grafted or just ungrafted rootstocks, the soil conditions, the management practices throughout the growing season all have a strong effect on the potassium accumulation. And so most of the studies that are published, they're doing petiole analysis during anthesis for interpretation of potassium uptake. And often juice or wine analysis is not reported. So at first I was really overwhelmed trying to, there was just so many stories to tell on different reports and results. So I really wanted to focus now on two articles that had extensive studies on different vineyards. And so the first I'll be discussing is Wolpert, which was published back in 2005. And so he did a study of 3 different vineyards, grafted to Cabernet Sauvignon, Zinfandel, or Chardonnay, but they were all grafted to 14 rootstocks. So even though it's 3 different vineyard locations, they had the same rootstocks, but different scions. And so I took his data of his 2-year study, his average data, and here in this graph, you can see on the y-axis is the Zinfandel total dry mass potassium percent, and on the x-axis is the Cabernet Sauvignon total dry mass potassium percent. So I regress, these 2 average values of the rootstock. So like, for instance, this is, where's my mouse? I keep losing it. 5Cs right here. And so I wanted to see, does the rootstock have a strong effect on the petiole potassium content, regardless of vineyard site location and regardless of the scion? And you can see that, yes, it appears that the rootstocks are having a strong influence on the potassium concentration in the petiole. This is reflected in the high regression value, R-square value of 0.6, almost 0.69. And to confirm whether or not this regression was significant, I did an F-test, and you can see the p-value for all 3 comparisons are much less than 0.05. So that suggests that, yes, these regressions are significant. And so Wolpert, when he published this data, that's all he completed was the petiole analysis, and that's what he used to interpret, certain rootstocks and the results of potassium accumulation. So the next article I like to present is by Walker, published in 2012, and he actually worked with Rule back in Australia in the early 90s, and this was more or less a continuation of Rule's work. And so he, he did 3 different vineyards, and at each vineyard he planted Syrah and Chardonnay grafted to the 8 same rootstocks. And so I took his data and for the petiole potassium content at anthesis, and I regressed the values. So you can see this, the Syrah is the y-axis, and that is potassium grams per 100 grams of dry weight. And then on the x-axis is the Chardonnay, same units. So I want to see, are we still, like Wolpert's data, are we seeing a strong correlation of the rootstock having a strong effect on the potassium accumulation? And so you can see here that, yes, it appears that You can see like at pathway, the regression was very strong with 0.78. The lowest was at Mirbian, which was 0.54. And so I also wanted to confirm whether or not these correlations were significant with the F-test. On the right-hand side, you can see the low P or below 0.05. So that represents, these are significant correlations. And one thing that was great about Walker is he didn't just stop here. He actually continued on through harvest and harvested the juice. So now I want to see, okay, are we seeing the same trends? Are rootstocks having a strong effect on the potassium concentration in the juice at harvest? And here you can see the regression values. Now it's Chardonnay as the y-axis and potassium millimoles per liter in the juice, and the x-axis is Syrah potassium concentration. So I want to see, are we seeing a strong correlation? And these values are, you can see the much not as strongly correlated. So the highest value was 0.39 at Corlong, at the Corlong vineyard. And so I want to obviously confirm whether or not this regression was significant. And you can see how the value was, the p-value is greater than 0.05. So it's suggesting that this regression is not significant. And that was the same, obviously, for the other two vineyards. So this was really interesting and especially for Rule and Walker, because that for years, so many articles have just been using the petiole potassium content as an indicator for juice at harvest. However, this extensive work by Walker in 2012 really shows that actually the correlation of rootstocks to potassium accumulation in the juice actually is much more complex. And whether it's scion is influencing the actual accumulation in the juice or harvest practices, something else is having an influence on the concentration of potassium. So now I want to ask the question, is really the petiole potassium at flowering a strong indicator for berry potassium content? So in the last 2 decades, very few reports have actually compared petiole potassium content to juice potassium content. Obviously, Walker and Blackmore in 2012 reported poor correlation, but also Zhao in 2020. He did a much smaller trial, and he also observed a poor correlation of petiole potassium content to juice potassium content. And so this really brings a big question to interpreting the potassium petiole content to juice potassium content. And so it's definitely a debatable topic. So potassium is remobilized in the phloem, and it's an adaptive and responsive system. So the amount of potassium that's going to accumulate in the berry is a function of the root uptake from the soil, the extent of redistribution from the leaves, and lastly, the mobilization of potassium from storage sinks. So for future research, they really need to have detailed analysis of environmental conditions influencing potassium uptake. A lot of the studies breeze over the soil conditions and don't really go into in depth of the pH of the soils, which we know can have a strong effect on potassium uptake. Also, identification of physiological, molecular, or genetic traits which restrict potassium uptake. As I mentioned earlier, 140RU appears to restrict potassium being transported up into the canopy. So being able to identify what genes potentially are restricting that uptake could be huge. And then also, lastly, research must include juice analysis of potassium concentration at harvest for proper interpretation. So for my acknowledgements, I really like to thank Professor Bartlett. She was my faculty mentor. She met with me countlessly through winter quarter, really directing me and giving me great advice as writing this literature review. I also like to thank Professor Cantu and all the other UC Davis faculty for my 2 years here in the program. Bennett Lane Winery for an awesome internship during COVID and also the fires. It was really difficult, but they ensured I had a great experience overall. The Drashville Stipend Award, and then lastly, most importantly, all my fellow colleagues that contributed to this great experience here at Davis. So thank you so much. Any questions? Right on. Well, if not, let's see, one. Thank you. Appreciate it. How are we doing on time? Are we doing good? Yeah, it was about 20 minutes. All right, well, if no one has any questions, Dar, should we continue on to the next presentation?
Yes. Go for
All
it.
right. So I guess I'll stop sharing my screen.
You have a question in the chat, Spencer. Can you use pH as a stand-in for potassium?
Yeah, so that's really interesting to ask. So I would say overall that a lot of articles do, but it is obviously just not only is potassium contributing to the pH of the solution, other cations are also going to be contributing. But yeah, I would say most of the research that is done on potassium accumulation is assuming that strong correlation between pH and the potassium concentration. So yes, I would say that I would be confident in using potassium. Yeah, thanks for the question.